Leden van het college van bestuur, collega-hoogleraren, geachte aanwezigen. Gaarne heet ik u van harte welkom in deze openbare zitting van de Senaat van de Rijksuniversiteit Groningen. Vanmiddag wonen wij de oratie bij van Dr. Prof A P Zhernakova, benoemd tot hoogleraar aan de faculteit Medische Wetenschappen, met de leeropdracht Human Genome and Exposome. Een oratie is de reden waarmee een nieuwe hoogleraar zijn of haar ambt officieel aanvaardt. Het is de gewoonte dat de oratie wordt voorafgegaan door een korte laudatio, waarin een daartoe uitgenodigde collega van de nieuwe hoogleraar de orator introduceert. Dat wil zeggen ons iets vertelt over diens, leven en loopbaan. Voor de introductie van collega Zhernakova geef ik graag het woord aan Dr. Nine Knoers, hoogleraar aan de faculteit Medische Wetenschappen met de leerstoel Humane Genetica. En verzoek collega Zhernakova aansluitend haar oratie te houden.
Honored Rector, dear colleagues, dear Maxime, children of Sasha, and other family and friends of Sasha. It's really a great honor and pleasure for me to introduce Professor Sasha Zhernakova, who today officially accepts her chair, entitled Human Genome and Exposome, by giving her inaugural lecture. Sasha was appointed full professor in July 2020. Sasha Zhernakova began her career as a pediatrician in Russia, where she worked as a consultant in medical genetics in St. Petersburg from 1998 to 2000. And in 2000, she moved to the Netherlands, where she started working in the molecular genetics laboratory of the Department of Genetics in the University Medical Center Utrecht, first as a volunteer. Just imagine having been a pediatrician and then starting here, wanting to do science as a volunteer. Some time later, she started working as a research analyst, but then in 2005, she met our current rector, Professor Siska Wijminga, and became her PhD student in the complex genetics group of Professor Wijminga. Sasha graduated cum laude with her thesis, The Autoimmune Puzzle, Shared and Specific Genetics of Immune-Related Diseases. And after 2 successful postdoctoral research periods at Harvard Medical School, Boston, and Leiden University Medical Center, respectively, Sasha started her own research group in 2012 in our Department of Genetics at the UMC Groningen, enabled by a Rosalind Franklin Fellowship from the University of Groningen. Sasha there initiated research on the so-called gut microbiome, the community of microorganisms such as bacteria in the gut. And she studied the complex interplay of environmental, microbiome and genetic factors in complex diseases such as autoimmunity and heart and vessel disease. Her analysis of the gut microbiome in participants of the multi-generational study Lifelines has led to several breakthrough discoveries. Sasha's group was one of the first to find strong links between gut microbiota and several human characteristics and diseases. And these discoveries were published in prestigious journals, such as Nature, Science, and Circulation Research, were highlighted as cover stories, honored as Best Paper of the Year, and or were the focus of lots of international media attention, all showcasing their outstanding scientific, medical, and societal impact. In recent years, Sasha's research extended to the longitudinal analysis of the microbiome in combination with environmental factors, genetics and other data in mothers and babies during pregnancy and in the first year of life of these babies. She does this in the so-called Lifelines Next cohort, a cohort of 1,500 mother-baby pairs. And the project's aim is to understand the combined effect of all these factors on short and long-term health in children. Sasha has achieved impressive funding for her research, including a prestigious VIDI grant from the Dutch Research Council, a competitive starting grant from the European Research Council, and as co-PI, a large gravitation award from the Dutch Research Council, on a consortium project aimed at studying the so-called exposome, the totality of environmental exposures in relation to health. Sasha has also received many scientific awards, among which recently the Gamov Award, established by the Russian-speaking American Science Association to acknowledge the Russian-speaking scientific diaspora for outstanding contributions to science. So altogether, Sasha is an outstanding and innovative scientist whose work has resulted in a paradigm shift in our view of the role of genetics-environmental interactions in complex diseases and trade. And she laid the foundation to support the gut microbiome as an important modifiable player in personalized medicine. Sasha is a very inspiring role model for young scientists. She strongly believes in team science, actively promotes open science, and accommodates students and colleagues from other departments to promote collaboration and harmonization of microbiome research. She's one of the founders of the Groningen Microbiome Hub, a multidisciplinary team of more than 40 PhD students and researchers working on various aspects of microbiome research. And finally, Sasha is a very kind and caring colleague who is interested in the work and lives of others, both within and outside her work. She is, impressively, very committed to helping the Ukrainian people in this terrible war and has opened her heart for Ukrainian refugees. She arranged a PhD position for one of them and offered several refugees a place to live safely in her home. Dear Sasha, I'm really, really pleased and proud that you are our colleague in the Department of Genetics of the UMCG. We very much enjoy having you with us, and we hope and expect that you will have a very successful continuation of your career with us in the coming years. For now, we're looking forward to hearing your inaugural lecture and wish you success in giving it.
Dear Rector, dear colleagues, dear friends, thank you for attending my inaugural lecture. It's an enormous honor for me to talk to you in this aula. Today in this lecture, I will focus on the fascinating world of the gut microbiome. For me personally, the topic of the gut microbiome represents one of my most long-standing scientific interests. In September 1991, I joined the junior scientific research community in the Department of Infectious Diseases in St. Petersburg State Medical University. The first presentation I made at this community was on the link of gut dysbiosis and diseases in children. The level of dysbiosis was determined in an old-fashioned way, as the ratio between beneficial and pathogenic bacteria based on the number of colonies that we grew from 1 gram of feces. Analysis of dysbiosis was already quite popular in that time, and it was a routine protocol for children with gastrointestinal complaints. Our analysis had concluded that gut dysbiosis is a signature of allergies, colics, and other infant diseases. Since then, I have considered the gut microbiome as an important component of health. In this lecture, I hope to share with you my excitement about the universe of the gut microbes. I will first talk about our current understanding on the role of the gut microbiome in health, and then discuss how we will follow up this knowledge in future. First, let me briefly introduce you to the microbiome. We are not living alone. There is a vast community of microorganisms living in our body and inside us. The largest community lives in the gut and is called the gut microbiome. This ecosystem is very complex and diverse. It includes bacteria, viruses, fungi, and other microbes. The total number of gut microbes is difficult to estimate, but we can be sure that there are at least hundreds of different species, thousands of different strains, and millions of microbial genes in every single individual. Altogether, the weight of the gut microbiome is about 300 grams, which makes it a large organ, bigger than a kidney, a bit smaller than a heart. When talking about the microbiome, we often mainly consider bacteria, but the main reason for this focus is that other members of gut community are less well studied. Researchers have been studying the gut microbiome for more than 300 years, since the discovery of bacteria by Dutch scientist and tradesman Anthony van Leeuwenhoek. For many decades, bacteria were studied in relation to diseases and were seen as pathogens. It was only about 100 years ago when the idea of good bacteria appeared. One of the promoters of this idea was Ilya Mechnikov, who thought that bad bacteria cause aging, while good bacteria can protect people from diseases. He considered bacteria from sour milk to be good and thought that drinking it could help individuals stay healthy. This was the origin of the idea of probiotics, the idea that introduction of good bacteria can promote health. Microbiome studies became particularly popular and successful in the past 10 years. The main reason for this is progress in technology. At the beginning of this century, methods for massively parallel sequencing were developed, and that made it possible to sequence DNA from a community of many different organisms at the same time. The technological advantage boosted microbiome research enormously and enabled wide-scale studies in large numbers of individuals. Altogether, tens of thousands of participants were included in these studies, leading to identification of thousands of microbes. It was about 10 years ago when we started the microbiome research in the Department of Genetics at the UMCG. Here, I would like to immediately acknowledge our rector magnificus, Professor Siska Weiminga, who in 2012 initiated the first microbiome collection in 1,500 participants from the cohort Lifelines Deep. In the same cohort, we also measured genetics, levels of proteins, levels of metabolites, and many other biological layers. The availability of this information allowed us to explore myriad relations between the gut microbiome and other biological outcomes. In 2016, a larger collection, now called the Dutch Microbiome Project, was initiated by Professor Martin Hofker. It included 10,000 participants, which at the time made it the largest metagenomic study in the world. These were very timely initiatives. They allowed us to perform pioneering studies in the human microbiome field. The very first PhD students started working on the microbiome data in our department in 2012, whereas now more than 30 members of the Groningen microbiome team are using the data generated from these two cohorts. Moreover, we made the resulting microbiome data available for researchers worldwide. They have been downloaded more than 100 times and used in various research projects elsewhere. The key factor in the success of these microbiome studies was the fact that they were done in collaboration with the cohort study Lifelines. For the tens of thousands of Lifelines participants, extensive health and environmental information is regularly collected. This allowed us to study the microbiome in combination with information of diet, lifestyle, environmental, and many host factors. The deepness of the data is truly unique, and I'm enormously thankful to all Lifelines colleagues and Lifelines participants who provide Groningen researchers a solid foundation for studying the variations in human health, including the microbiome. So, what did we learn from the past 10 years of microbiome research? First, we learned that the individual microbiome is enormously variable. As humans, our genetic material is 99.9% alike. For gut bacteria, the overlap is much smaller, with only 10% of bacterial composition shared across the people in this audience. When considering the gut virome, this situation becomes even more complicated. The virome is very individual specific, with only a small number of viruses shared across individuals. This high variability leads to the fact that the majority of microbes are rather rare and still not well characterized. Second, the microbiome cannot be disconnected from its host, the human being. Almost everything what we humans do has an effect on gut microbiome. Food, sport, sleep, lifestyle, medication, diseases, everything matters. This includes not only obvious factors such as diet or current lifestyle, but also family history and early life factors. For example, we see long-standing changes in the microbiome depending on the place where someone spent their childhood, whether it was in the city or in the farm. We also see an effect of our genetics on the abundance of some bacteria. The third important observation is that even with the extent of the individual variations, we do see some common pattern of bacteria that are related to health. When analyzing the associations on the microbiome with different diseases, we often see that some bacteria are increased or decreased in unrelated conditions, such as diseases of the gut, metabolic diseases, cardiovascular diseases, mental health, and others. The picture is, however, not black and white. It is important to consider that for different individuals, the same microbe or pathway can be either beneficial or rather pathogenic, and this effect can also depend on dozens of factors. Still, the existence of the disease characteristic bacterial pattern is an important and positive observation. We can now focus on the community of bacteria that are generally beneficial for health and on the other set that we would rather want to eliminate from the gut. Finally, the microbiome is not just associated to disease. In several cases, we can now also prove its causality. For example, fecal microbiota transplantation in mice is a common method for testing causality. In this practice, fecal material from individuals with certain condition or disease are given to germ-free mice who do not have their own microbes. By comparing the outcome of fecal microbiota transplantation from different donors, healthy or diseased, we can see the effect of individuals' microbiome on such traits as metabolism or weight or behavioral traits, for example, anxiety, or even response to treatment, for example, the effectiveness of anti-cancer medication. Often we do not understand exactly how changes in microbiome affect health. But there are some examples when we can identify the mechanisms. We know, for example, that many metabolites in our body are determined by gut bacteria. For example, some bacteria can produce short-chain fatty acids, which are considered good metabolites. They can improve glucose metabolism and regulate our immune system. In another example, a set of gut bacteria are involved in metabolism of tryptophan to serotonin. Tryptophan is an amino acid. We get it from food. Serotonin is a molecule that is involved in learning, memory, and happiness. This illustrates a direct link where the gut microbiome is a mediator between our food and our mood and brain function. So these are a few highlights from our current understanding of the microbiome. The next question is, where do we want to go? What do we want to achieve? And how? It is my strong belief that we should aim at eventually using the microbiome as a target to prevent or treat diseases. In the next part of my talk, I would like to highlight a few research directions that we will follow to achieve this goal. It is good, however, to realize that these directions will not always be straightforward, but might rather look like the Strava images, the running map images from the orienteering competitions that we used to run together with my husband, Maxim. So first point to go. We do not yet understand the whole complexity of the gut ecosystem. Even after analyzing more than 10,000 Lifelines participants, we keep identifying novel bacterial species. Even more importantly, our current characterization and understanding of the gut virome is very poor. The research community has constantly discussed viruses in the past 3 years, but our interest was mainly in the human viruses in relation to COVID. There are also millions of viruses in our gut, but most of them are viruses that infect bacteria, called bacteriophages. Virome analysis faces enormous challenges. The viruses are, in general, much more diverse than any other kingdom of life. They have a high mutation rate, which means they change fast, and they do not have good marker or signature gene that we can use to characterize them. Viruses can also get in and out of bacteria. They can stay there silent for a long time. They can transfer genes from one bacterium to another, or they can kill bacteria. The actions of bacteriophages and the logic, what exactly dominates their behavior, is poorly understood. On the other side, it's important to realize that bacteriophages have large therapeutic potential. Bacteriophage cocktails for treating bacterial infections were first proposed almost 100 years ago, before the discovery of antibiotics. A lot of work in this field has been initiated in Tbilisi, capital of Georgia, and I was fortunate to study the opportunities of bacteriophage therapy during my medical education. Recently, a pioneering study of the international group of researchers used a cocktail of bacteriophages to eliminate pathogenic bacteria as a treatment for inflammatory bowel disease. We can expect that many more such studies will appear in coming years. So, deep characterization of microbiome, and in particular the microvirome, is one of the tasks for future. Second, it is important to realize that humans and microbes form one joint ecosystem, and the microbiome should be studied in combination with host factors. Host factors can be split in two. On one side, our genetics, which is stable and never changes. On the other side, we experience a constantly changing environment, the 100 of environmental factors around us, which together are called the exposome. If we want to determine if microbiome can predict the development of diseases, and that's what we obviously want to determine, we need to monitor the microbiome in pre-diseased individuals. We should follow them longitudinally, simultaneously tracking other factors that contribute to well-being, including genetics, lifestyle, diet, diseases, and many others. This all sounds like a huge and ambitious task, and it actually is. But we have already started making it a reality. In Groningen, we are in the leading position for this research due to the unique opportunity to join forces with Lifelines. It means we can sample stool from participants who will be followed for decades. This would allow us to later go back to samples from before the onset of a disease and investigate the microbial signatures of the future diseases and explore it in combination with environmental factors and their changes. It is of ultimate importance that the Lifelines initiative continues for coming decades because it is essential for performing comprehensive studies on various aspects of health, including the microbiome. I expect that in a few years from now, these longitudinal studies will bring us to a knowledge of using microbiome as biomarkers. This knowledge can be used in the same way as we now use it for other well-studied biomarkers. For example, we can routinely measure blood lipids in combination with host genetics and lifestyle factors such as smoking, and by putting these factors together, we can quite accurately predict the individual's risk of developing cardiovascular diseases. In the same way, I expect that we will soon know which changes in specific gut bacteria or viruses in combination with lifestyle, specific diet, and genetic factor can substantially increase or decrease the risk of a specific disease. This knowledge will be very practical because changing the gut microbiome, though not very easy, is still more realistic than changing other parameters, such as genetics. I also expect that in future, the methods for sample collection and analysis will be easier and more popular. Collection of stool is non-invasive, and technological development suggests that even home devices, such as smart toilets, will be able to sample and profile microbes. Collecting extensive environmental factors is much more challenging due to their high number and variations. But lifestyle already has a globally unique exposome database, and on top of that, information on the exposome can now be collected by various devices, such as smartphones for measuring activities, dust boxes for analysis of local environment, and wearable rings for collecting metabolite concentrations around us. Many corresponding methods are now being developed by members of the Gravitation Consortium Exposome-NL. I would like to thank all the participants of the Exposome-NL Consortium for their successful collaboration and joint projects. I hope I convinced you that longitudinal collection of the microbiome, in particular in pre-diseased individuals, is essential for understanding the individual-specific host microbiome relations. But of course, if you want to measure all the important environmental and microbial changes that can influence individual health later in life, we have to start early. It would be best to analyze microbiome samples from the preconception period, then follow parents during pregnancy and their children from first day of life. This is a dream project that brings together my background in pediatrics and my current interest in microbiome and exposome. And I'm very happy that I can focus my research on such a project, which is Lifelines Next. Lifelines Next started as the fourth generation of lifelines. In 1,500 families, the microbiome, questionnaires, and many other sample types are being collected from mothers during pregnancy, from babies and mothers at birth, and regularly during the first year of life. The overall aim of Lifelines Next is to explore the role of prenatal and early life factors on children's health. There are many specific questions that we aim to answer in the Lifelines Next study. For example, which pregnancy and delivery factors are crucial for the long-term well-being? Or, how exactly do maternal diet or parental smoking influence babies' health? Or, also importantly, why do some babies cry all the time? Of course, in my research team, the special focus is on the role of microbiome environment in developing and mediating these conditions. We study not only the gut microbiome, but also oral, breast milk, and other ecosystems. The inclusion of mothers and babies into this cohort is still ongoing, but we are already making exciting observations. For example, we can now explore how exactly the gut ecosystem develops in babies, what are the origins of gut bacteria and viruses, which important bacteria and nutrients are coming from breast milk, and many other research questions. There is still a lot of work to do, so this is just the beginning of many exciting studies. Lifelines Next is a huge project initiated and supported by many people. I would like to give my deep thanks to several UMCG departments and lifelines who started this project in 2016, to the UMCG for providing initial funds, to the core group and steering committee for their support, and to all nurses and our management team. I also thank UMCG Cohort Hub for recognizing Lifelines Next as one of the priority cohorts of UMCG and providing funds for data and sample management support. But most of all, I would like to thank my research team working on Lifelines Next. It's your enthusiasm, curiosity, creativity, hard work, and critical attitude that makes Lifelines Next research successful. I was talking about comprehensive longitudinal studies, which are enormously important. But it is good to realize that even after completing them, we would still not be ready to use microbes to prevent and treat diseases. To make microbiome-based medication a reality, we need to understand the functional properties of each microbe and how exactly bacteria and viruses are linked to health. We also need to develop medications in the form of specific microbes of their communities or metabolites or prebiotics. In the field of functional characterization of microbes, we can compare our current position with the early days of genetic studies. Genome-wide association studies in genetics became operational around 2006, and in just 10 years, we identify hundreds of genes that are associated to common diseases. However, despite years of work, the progress of characterizing function of associated variants is still challenging. Now we face a similar challenge in the microbiome studies. Association analysis is rather straightforward, whereas functional implementation is slow and requires a lot of practical work. Here, again, the UMCG provides an excellent research environment, as several advanced methods for functional analysis are already operational here. One research line is bacterial culturomics, the methods of culturing single bacteria and its further characterization. The group of Hermi Harmsen in the Department of Microbiology has huge experience in culturing both aerobic and anaerobic bacteria. By running culturomics in a collection of gut bacteria from patients and from healthy individuals, we can further characterize bacterial strains to understand the mechanisms of association of these bacteria with diseases. Specific microbes can then be applied to model systems, such as the organ-on-a-chip model, which is another strength of UMCG. Organs on a chip, including gut and liver on a chip, have been successfully developed in our department by several scientists from the Netherlands Organ-on-a-Chip Consortium. This model system effectively mimics the human intestinal tract. Adding single microbes or a community of microbes or microbial metabolites to this system will allow identification of their effect on gut and liver tissues, on intestinal permeability, and on the immune system, and will enable direct measurements of bacteria-bonded metabolites. Culturomics, organs on a chip, and other functional methods will definitely be the follow-up of future microbiome work. It is clear that zoom-in studies into microbial functionality is a large task that requires joint forces from scientists from different disciplines. It is therefore a great achievement of the whole microbiome team and a big recognition of success from UMCG that we were given the opportunity to create a new facility, the Groningen Microbiome Hub. The hub is still under construction, but will become operational already in April. It will combine work for large-scale metagenomic analysis with culturomic studies, organ-on-a-chip work, and other functional studies. This is a big success of many researchers from the Department of Genetics, Microbiology, and Gastroenterology. And I also would like to acknowledge the UMCG board, who recognized the importance of microbiome research and supported this initiative. I'm sure opening the Groningen Microbiome Hub will bring our microbiome research to a new, deeper, and functional level. It's also important to realize that this achievement is just the start of the new level of microbiome research. It will still require a lot of clinical and research work, new investments, and collaboration with other researchers and industry. The final point that I want to discuss is how to bring all this information to patients. The Lifelines Next and Dutch microbiome projects have already generated a tremendous amount of data. The extension of this project to large sample size, more wearable devices, and more phenotypes will bring the amount of data to a much higher level. The next challenge that we will face is interpretation of the data and bringing it to patient care. In fact, translation to patient care is already happening. A few years ago, I gave a lecture on the microbiome in a scientific forum in Tartu, Estonia. After the lecture, one participant came to me with a question. She said, "I have 3 kids, and I made a gut microbiome analysis for all of them. 2 of them have high abundance of Bacteroides, which is one type of gut bacteria. The 3rd child, on the contrary, has little Bacteroides, but a lot of Ruminococcus, which is another type of gut bacteria. What can I do with these results to make sure my kids grow up healthy? Should they eat a different diet? Which one?" This is and will for a while remain a difficult question. Interpretation of an individual's microbiome in relation to health is a narrow path between Sil and Charybdis. On one side, it's very easy to overinterpret the current knowledge without a clear understanding of the downstream effect of specific microbes. On the other side, it's probably unrealistic to wait until we will get a full understanding of the microbial complexity. General advice based on our current knowledge, in principle, can already be helpful. For example, we know that on the population level, a diet rich in vegetables, fruits, and fermented products is on average associated with positive signatures of the gut microbiome. We can recommend this diet to almost everyone. Here, again, we're in a similar situation to the one genetics was 10 years ago, where interpretation of the genetic risk of common diseases was constantly balancing between valuable associations and low confidence predictions. In genetics, we are now getting much better by applying polygenic risk score that is based on the analysis of hundreds of thousands of individuals. For the microbiome, I expect similar trend, and this process could go faster due to technological and computational advantages. On the positive side, there are already several examples of more personal implementation of microbiome analysis. One notable example is the personalized nutrition project from the Weizmann Institute, Israel. Researchers there first identified a link between individuals' microbiome and their sugar metabolism. Next, they used artificial intelligence to develop an algorithm based on individuals' microbiome profile that allowed the prediction of the reaction to blood sugar to various food. Finally, they developed a service where participants can get recommendations about the optimal diet to maintain their blood sugar levels based on their gut microbiome. Application of this algorithm in pre-diabetic patients showed that participants could keep their blood sugar in a healthy range and that it worked remarkably better than just following the general recommendations of the American Diabetes Association. These and similar studies are current examples of using the microbiome in preventing diseases. This study is also a great example of using AI in the interpretation of microbiome results. This is the field that will be massively developing in the coming years, as we can already see in the examples of ChatGPT, DALL-E, and other tools. Introducing the microbiome into medical care will require training the new generation of researchers and medical doctors. For medical doctors in particular, knowledge about the microbiome is as important as understanding the function of any other human organ. The microbiome, its function, its relation with immunity, metabolism, diseases, and environment, and the interpretation of the individual's microbiome should be part of the basic medical curriculum. The Groningen Microbiome Hub can and should play a role in developing microbiome-focused courses for researchers and medical students. I am proud to say that our work has already made a notable contribution to education. Biology books for the 5th year of Dutch high school include chapters describing the basic of microbiome studies with illustrations and the text based on our results from the Dutch microbiome project performed in the Lifelines population. I believe this is a true recognition of the success of our team. In the last 30 minutes, I have discussed the enormous progress and success of the microbiome field, to which the researchers of the Groningen Microbiome Hub have made significant contribution. A lot of exciting work is ahead, and this work requires good planning, proper training, extensive collaboration, and, most importantly, efficient teamwork. I am enormously thankful to all my colleagues from Groningen Microbiome team for being active, creative, and critical. You are a great inspiration for me, and I learn a lot from you. I also constantly feel I'm a very lucky person to be able to work with the best scientists ever. When we first came to the Netherlands, I did not have a work permit and was looking for a volunteer job. I came to a genetics seminar in University Medical Center Utrecht and randomly approached one of the attendees. This person was Siske Wijmenga. What luck! I very much appreciate all of Siske and Martin's support and trust since then. Also, my enormous thanks to Jinguan Fu and Rinse Weersma for our pleasant, joyful, and successful collaboration. My path to this stage was not straightforward. Moving my career from clinical geneticist to volunteer and lab technician and then to researcher and group leader, a journey that took place in 3 countries with 3 children, it would not have been possible without the enormous support of my family and friends. I would like to thank my kids for their curiosity and for our scientific discussions, my partner Maxime for support and for bringing practical and realistic suggestions when I missed them, and most of all, my mother and my mother-in-law, who gave me a lot of support by taking care of the kids, sometimes literally passing them from one hand to another. Without you all, this achievement would never be possible. Thank you. Ik heb gezegd.